I am Kevin Lafferty. I'm a marine biologist and parasitologist. I have been working out of the University of California, Santa Barbara, where I work for a federal science agency called the United States Geological Survey. You might have read in the news that we have a new president, and as part of that, we have had some restrictions on our ability to travel, which is why I was not able at the last minute to join you in person. Literally, just a couple days before I was going to get on the plane, we learned that we could not travel. Um, so hopefully sometime in the future, I'll get to meet you guys. And I just wanted to say that if you find what I am talking about relevant to your own work, don't hesitate to email me. I'm eager to talk about parasites and food webs and lots of other things as well. So yes, parasites and food webs I think it's just a conceptual framework that I have found really useful to help group a lot of different research projects that my lab and I have worked on over the years. And in particular, the focus of this uh, uh, working group that you guys are doing is, I think, so exciting. I think there's so many fascinating things to do in this field still. And I wanted to really go with maybe sort of a larger context of how the things you're talking about fit into other things on a bigger scale. But what I decided to do was to select out some of the work that we've done that focuses on what happens when parasites actually get eaten in the food web. And so I want to start with a little video about some of the things that like a typical day in the field for our lab in this video here. So we're, like I said, marine biologists, and not sure if you can see the video well, but we're out deep sea fishing for this beautiful fish called an ono. And then when we dissect our host, we're very happy to find all these lovely parasites. This is a large trematode worm. My PhD student, John. And I want to say a few things about these parasites. They are probably they have one of the most admirable life cycles lifestyles that I can imagine. They are usually found in the stomach of this fish. They're large. They live for many years. They're constantly feeding on the blood of the fish. And there's almost always two of them, and they're almost always permanently copulating. So they're basically just for their entire multi-year life, just having sex and eating constantly. And I think it's one of the most romantic things in nature. And you notice how big they are. And of course, in the theme of this particular group today, I just want to say that for a little garlic.
Oh no! Oh no! Oh no!
So we have 1, 2, 3, 4, 5, 6, 7. Do we have any garlic? Look at that one. You got the sucker right there.
Remember to never go up for confidence. Waiting for that. Well, you don't know what you get tonight. So.
Anyways, we're allowed to do that to our graduate students in the United States. Right. But I brought this up because the whole idea about eating parasites is it opens up this huge number of interesting questions and ideas and potential for population dynamics and so forth. And so, of course, you should all probably be aware of this particular aspect, the zooplankton, nutrient, phytoplankton food web that is just so critical to our entire globe dynamics. And then the increasing realization that we have these chytrid parasites in the system and that they have the potential to greatly affect the way that these food webs work. And as I'm sure you've already discussed, phytoplankton basically come in two flavors, the ones you want to eat and the ones that you can't, if you're a zooplankton, and chytrids help solve that problem for zooplankton by changing inedible phytoplankton into edible spores that zooplankton can eat. And the complexity of this web, I mean, look at all those arrows. Opens up a lot of possibilities for different theoretical outcomes, different outcomes at different times of the season and the lake and so forth. Because I think you guys have talked about this concept a lot already, I just want to point out that this particular complexity that we see in lakes with respect to parasites and food webs is really just one part of a very complex lake food web. This is from Takvatn in Norway. And all the red arrows in this food web diagram are host-parasite interactions. And that chytrid is right there in the lake. So it's just one of many, many, many host-parasite interactions in a lake food web. Of course, I happen to think it's a very important and a very exciting one, but there are also other important and exciting food web links in this particular lake. I got interested in this topic a long time ago. Some of you may not have been born, I'm not sure. Back in 1997, some colleagues of ours from Canada, David Marcogliese and David Cohn, published this paper. And you probably know, if you know Canadians, they're very nice people. They are very polite. They also have the unfortunate geography of living in a country right next door to the big bad United States, so that they always feel a little bit underappreciated by us American scientists. And so they had a complaint, and they made their complaint very politely when they wrote "Food Webs: A Plea for Parasites." And the main point that they were making is that ecologists had published literally hundreds of food webs, and that the food web was a foundational concept in ecology. It underpins so many things that ecologists were thinking about. And yet ecologists never really put parasites in food webs. And they said, hey, ecologists, there's a lot of parasites. And sometimes these parasites may be doing important things, and we think you should put them in food webs. And so at the time, we had been doing a lot of work on parasites in a very small estuary, the Carpinteria Salt Marsh. It's about 20 minutes from our campus. And it was a very convenient place because it's a nature reserve for our students to go and do their projects and so forth. So we knew a lot about that system. And one of the things that we appreciated about the estuary was that it's not very diverse. There's not too many species in that system. In fact, there's only 39 birds, 8 fishes, 29 invertebrates, and 12 basal taxa. And so we thought, well, this is a good system where you could build a food web, because frankly, at least conceptually, building a food web only requires two things. You need a species list like this, and then you need to figure out the feeding links for who eats those species. Now, the reason that we do that is even an estuary is a very complicated place. When you walk out into the field, there's so many things going on. But we would like in science to make simple mathematical constructions of nature so that we can understand it differently, both theoretically and quantitatively. And so a food web, again, is just it's an incredible oversimplification of what this complex system is supposed to look like. And so when we put those feeding links together and we arrange them by trophic level, that simplification of the food web looks like that, which of course is not very simple at all. It has a lot of complexity, but the value of it is we can start to visualize what that food web looks like. And we can also compute some mathematical properties for it. Another way to look at that food web is to put it in a matrix where every species is listed as a row and a column, and a link between a predator and a prey is represented by a dot. So predators are the columns and prey are the rows. And so, for instance, Here we have a snail, which as a predator, it eats some diatoms that live on the mud, and as a prey, it's eaten by crabs and birds. So this is how we would typically an ecologist would put a food web together. But of course, we studied parasites, and in that system at this point in time, we knew that there were about 44 common parasite species. And so to integrate them into the food web, we start with our free living food web, and then we overlay the parasites on top in the red color. And what we see is, as in that lake food web, the parasites really change a lot of elements of the structure of the web. Obviously, they add a lot of diversity to the web, but they change certain characteristics. So for instance, let's go back to this matrix example of the food web, and let's add the parasites in. And now the top left square is what I showed you before. This is the classical, typical food web that ecologists have built for decades. And we also have parasites that eat hosts. So this is what you call the parasitism box of that food web. As soon as you start adding these in, you see, wow, there's a lot of links, as I mentioned before. But it also reveals that there's these other aspects of the food web that we hadn't really even thought about. So for instance, some parasites eat other parasites, and that's this box here. And then finally, and one of the things that I'll probably be focusing on in this talk, is there's this third box that nobody really thought about, and this is predators that eat parasites. And it turns out there's more links in this box than all the other boxes individually. So this is one of the reasons that parasites dominate food web links, is because they're eaten quite frequently. And so one of the questions, of course, is, well, how important are parasites in food webs? And then secondarily, what are some of the things that happens to food webs when parasites are eaten? But before I get into that, I wanted to address a common critique that ecologists have had of our work in the past. And that is, well, yes, you have a lot of parasite species and they have a lot of links in food webs, but they're small, and so maybe they're not that important from an energetic perspective. And so one of the ways that ecologists evaluate energetics of ecosystems is through these simple allometric plots, where you can plot the body mass of an organism against its abundance. And in this figure, these are species in our estuary food web. The birds are here in red, the fish are in blue, and the invertebrates are in green. And you can see that they all fit, at least on a log-log plot, nicely on this linear slope that has a -3/4 slope. That's one of the key theoretical outcomes of this type of work, and it's explainable by the fact that small organisms have higher metabolism rates, so they burn through energy a lot faster, which means they can't be as abundant as an equivalent organism that has a larger body size. Larger body size organisms are a lot more efficient. So for instance, if you have an elephant, which is a big body mass, and you were to chop it up into mouse-sized pieces and let those mice run around, those mice would use a lot more energy, and you wouldn't be able to maintain the same amount of mouse biomass as elephant biomass. That's just the underlying theory, but the point that we want to do is, what happens if we put parasites on this graph? Because nobody had ever put parasites on this type of graph before. If parasites are unimportant, then they should fall over here in this lower left-hand corner. And to a certain extent, they do. So parasites are not as abundant as free-living species that have similar body sizes. Obviously, they're smaller than free-living species, but they're not as abundant as we would expect if they were free-living species. And we looked at the one conclusion from this would be to say that, yes, ecologists are right. Parasites are not important in these food webs, energetically speaking. But another possible conclusion is that parasites have been wrong about the entire theory. In other words, that this allometric scaling plot is missing a very important key variable, and that variable is the trophic level of the species that are involved in the ecosystem. And so, for instance, if you go out to the Serengeti, you'll see lions and zebras, and they're about the same body size, more or less. But there's a lot more zebras in the Serengeti than there are lions, primarily because they are herbivores and they're able to support a much higher total population size, obviously, as an herbivore than as a top predator. So when we statistically control for trophic level in these plots, we rearrange the data, and what we find is that parasites and free-living species all play by these same three rules. This relationship is universal amongst parasites and free-living species. Parasites are every bit as abundant as free-living species once you consider their body size and their trophic level together. This helps account for where parasites fit in ecosystems. By the way, controlling for trophic level also makes these plots a lot better. So not only did it sort of, I think, help ecological theory in general, but it added and controlled for a lot of the noise in those allometric scaling plots. I wanted to give you an example from the estuary in terms of the biomass density of species that are in that system. And so let's start with a group of species in that estuary that we know are important in the food web. These are the shorebirds and other birds that live in the estuary. And what I'm doing is I'm plotting birds in a style that maybe a chicken farmer would plot them, which is kilograms per hectare. It's not usually the way we think about birds in nature, but this is their body mass density. And we see that in the winter, we get a large influx of migratory birds coming into our system. And there's 2 to 3 kilograms per hectare in our system. If we compare this with another community, in this case, a community of parasites, the trematodes that infect snails, what we find is that there's a lot more biomass density of this parasite community in our system than there are birds. So if we were to consider that birds are important members of this community, then we might also, by this measure of biomass, consider that parasites are as well. Okay, so I want to talk a little bit more about trematodes. They're the main group that I study, and they have these famously complex life cycles. And in the estuary, the most common group live as adults in birds. And then they have eggs and larvae that pass out of the bird onto the mudflat, and they infect a first intermediate host snail. And in that snail, they develop as a rhedia form. And I spent most of my dissertation working on that interaction, which is I won't talk about today. But what they do is to go to the next host, they produce a free swimming stage. And in many ways, it's like a much larger version of a chytrid spore. And it goes out, and instead of looking for another snail, it looks for a different second intermediate host. Or it might be a clam or a fish or a crab. In this case, we're going to talk about fish. And then the life cycle is completed when a bird eats an infected second intermediate host. Now, one of the things that I wanted to mention is that I said that these trematode cercaria are kind of like chytrid spores in a way. And so many years ago now, we wondered, does anybody eat these things? And that's because they are, they look like zooplankton. Basically, they are part of the zooplankton community. And in some places, they actually dominate the zooplankton community in these estuaries. They're not protected. They don't have defensive measures. They don't have toxins. They have lots of glycogen. They live for about, I don't know, maybe 24 hours maximum, generally less. And so we did little experiments where we looked at, in this case, 7 different species of trematode cercariae, and they differ by body size. And the size of their bodies is represented by the size of those points. And if we plot the size of the parasite versus how likely fish are to eat them, what we find is that no fish eat the littlest ones, And everybody loves to eat the biggest cercaria. So as in a lot of studies on trophic dynamics, the size of the prey is really important in terms of determining who gets eaten. Now, I wanted to point out two interesting things about the natural history. The first is that this little tiny guy that nobody eats, we'll talk about him in a minute, he infects fish. And so maybe it's not too surprising that fish aren't trying to eat him. But this species, the trematode that is most eaten by fish, infects fish too. In fact, that trematode mimics food. It groups together to form this big mass. It's brightly colored, and it acts like a large zooplankton or even a polychaete worm or something on the bottom of the estuary. And fish eat it more than anything, and then it infects the fish and lives in the fish's liver. So in this case, we have, for all these species in the middle, when they get eaten, they die. But when this species get eaten, it completes its life cycle. So the consumption of these stages by fish appears to be important in the food web. Should also mention that it's the little fish that are more likely to eat cercarias. So big fish don't really find cercaria that interesting. It's not in their size range in terms of they don't see it much as a food, but the little fish, they love to eat cercaria. So we've done the energetic calculations to show that cercaria in the system probably comprise 10 to 20% of the energetic budget of at least the small fish species in the system. Now, remember that parasite I mentioned, that the little one that infects the fish. I want to talk a bit more about that because it adds an additional twist to our system. It's the most common trematode species, and when it infects the fish, it moves through the circulatory system, and it ends up coating the brain. And the brain of this fish is covered by hundreds of these baby parasites in a larval stage called a metacercaria. And if you recall, the next stage of this parasite's life cycle is in a bird. So anything that this parasite can do that can increase the chance that the fish will be eaten by a bird should be under pretty strong selection. And you can imagine living on the brain of your host is a very powerful place to be if you want to manipulate it. And indeed, when you take the fishes into the lab, they exhibit these conspicuous behaviors. You can also see these behaviors in the field. Hopefully this video will play for you. The video is playing very slow on my end, but really the point is that the fish are pretty cryptic and nondescript, but once in a while when you watch a school of them, you'll see a fish will roll over on its side and it'll flash that silvery belly. And I'm not really seeing it great there. Once in a while you can see one flash. And then the birds are going around and they're feeding on fish. And the question is, are they feeding more on infected fish? But first, let's ask, do the infected fish behave differently? So when we look at fishes in the laboratory and we count their behaviors, what we find is that uninfected fish over here, this is infection levels on the x-axis. The uninfected fishes display a few conspicuous behaviors, but the infected fish display a lot more, and the heavily infected fish display the highest frequency of conspicuous behavior. So something about having parasites on your brain changes your behavior in ways that may make you more conspicuous. And when we then ask if the birds actually eat more of these infected fish, we can assess that with a field experiment where we can go out into the estuary and put fish in these pens and cover one of the pens with netting to keep birds from foraging on the fish. And then birds will go in and eat fish in that open pen eventually. And by comparing what types of fish were eaten in the open pen with the control pen where birds could not enter, we can deduce that almost no uninfected fish are eaten by birds. Almost all the heavily infected fishes are. And to get this result means that infected fish are 10 to 30 times more likely to be eaten by birds. So this is an example of how, in this case, remember, the parasites are being eaten by the predator, but they're actually in control of that predator-prey dynamic. They're determining who lives and who dies in that predator-prey system and are potentially driving some of those predator-prey dynamics. Now, I have focused so far on our work in estuaries. I showed you earlier that Tokvatten food web, and I just wanted to point out that everything that I'm saying about the estuary is similar for other systems that we've looked at. So we've gone out to Palmyra Atoll, which is a isolated coral reef out in the middle of the Pacific Ocean. And when we build our food webs there with parasites, we find that they look very similar, both visually and mathematically, to the ones that we build in the California estuary. So I suspect that in many places that we look at parasites in food webs, we will see that they dominate food web links. We will also see that one of the most common types of interactions in these food webs are predators eating parasites, either the free living stages of parasites or parasites that are in the host that the predator feeds on, and that in a large proportion of these cases, in the estuary anyways, it's about a third of the times when a predator eats a prey with a parasite in it, the parasite can infect the predator too. So obviously parasites are in food webs and they've evolved to be in food webs, and they exploit the food web in many cases, potentially by manipulating the behavior of those prey. I want to show a video that is one of the most extreme examples of manipulation, and hopefully it will play for you. This is a cricket in France, and it's up by the swimming pool. It's going to jump into the swimming pool. And the reason that it jumped into the swimming pool is because it's infected by this larval nematomorph worm, which is going to emerge as an adult and go off to try to find mates in the aquatic system. You know, these are fascinating parasites. We love showing this example as a marvel of evolution, and et cetera, et cetera. But in so many cases, when we give these examples and we lecture and so forth, we show them mostly as curiosities. We tend to think less about the fact that they might have actual ecosystem level effects. And so then the next study I want to show you is from collaborators with Japan, which I think is probably the best empirical example of parasites having energetic effects at the ecosystem level. And this work happens in these small streams in Japan that have been impacted by habitat degradation, deforestation, and overfishing. And that has led to the decline of a fish in these Japanese streams called the Kirakuchi char. And my colleagues are they're fisheries biologists, and they study this endangered species very, very carefully. So they go to these streams, all the fish are tagged, use electroshocking to capture them twice a year. They're able to measure their reproductive status, their weight, their growth. They pump their stomachs to see what they're eating, and because they care about these fish. And one of the things that they find is that these fish eat what you would expect. They eat the insects that live on the bottom of the stream, the benthic insects. That's a big part of their diet, at least most of the year. In September and October in the fall, when they pump their stomachs, they find that the trout are full of crickets. And what they eventually figured out was that these crickets were jumping into the water, and they were infected with those nematomorph worms like the one I showed you in that video. The cricket is obviously swimming around helplessly on the surface of the water, and it makes a very easy food source. And even though the parasite has taken a lot of energy from that cricket, there's still plenty left for the fish to consume. Now, because they've been doing all this careful energetics and diet work, they were able to assess the energy budget of these fish throughout the year and assign it to different sources. And what they discovered, I thought was pretty amazing, which was that, yes, the fish did eat benthic insects. That's about 20% of their energy. They also eat terrestrial invertebrates that fall into the stream, about 22%, but 60% of their total energy budget is delivered to them in the form of crickets that are manipulated by this parasite. This has additional food web implications. So they did this incredible experiment where they were able to set up barriers to prevent crickets from jumping into the stream, and then they could measure aspects of the food web. And so here's basically what the food web in the streams looks like without parasites, when you keep the parasites out. And the arrows show the strength of the energy flow through that system. But normally when the parasite is in the system, this is what those arrows look like. And the main point I wanted to mention here is that the fish, instead of eating the insects that live in the stream, they're eating the crickets. And that releases the benthic insects from predation by the trout. That means more benthic insects. That means more grazing on the periphyton. That means different effects on nitrogen uptake. It means different and more leaf breakdown rates and so forth. So there are cascading effects of this particular dynamic on the stream food webs. And they make the point, and I rather agree, that it might be difficult in some cases for this endangered species to persist without the parasite delivering it 60% of its energy budget every year. Well, this whole idea about trophic cascades and food webs, I think, is really fascinating about these dynamics and feedbacks that happen due to predator-prey interactions. And so I wanted to give you just one example from our work, and this is looking at the Galapagos food web. This is a very simplified version that shows key players in a trophic cascade that include humans. So humans are also often involved in trophic cascades in aquatic systems. So fishermen eat hogfish, which eat sea urchins, which eat kelp. And so in areas where you have a lot of fishing, you have few fish, you have lots of sea urchins, and you have little kelp, compared to some of the marine reserves that are in the Galapagos that have lots of fish, few urchins, and a lot of kelp. And one of the reasons that I was interested in this system was a PhD student had contacted me about the parasite that he was studying here. It was actually two species of parasitic snails. So a whole group of parasitic snails called the eulimids in marine systems. Two species here. This one makes little galls in the tips of the urchin spines. This one sticks its proboscis through the test of the urchin and basically sucks out the gonads. It's like living in your own milkshake, basically. And the one that sucks out the gonads has pretty substantial fitness effects on its host. So if you plot the amount of gonad that's in a sea urchin against the number of parasitic snails that it has, you can see there's a very strong decline. If you have 5 parasites on you, you have almost no gonad left. So the reason that a food web context is important here, and especially feeding on parasites, is that there are little crabs, these decorator crabs, that are associated with the sea urchins, and their favorite food are the parasitic snails. And so if you find a sea urchin without a crab, it tends to have, on average, a parasite on it, at least one. But if there's a crab present on that urchin, you very rarely find a parasitic snail on it. So the crabs do a good job of eating the parasites from this parasite. Now, the crab doesn't eat the sea urchin, it just is using it for shelter. So it's a real complex setup. And when we then put this in the food chain context, what we find is that there's a lot of positive and negative effects on this system that involve the parasitic snails. And when we ask, what is the net effect of fishing? What's the indirect effect of fishing on parasitism in this system? It's very difficult to deduce from this food web what fishing does, because the food web is complex enough. It's not a linear food chain that we cannot make simple predictions. It turns out that by comparing, when we go to marine reserves and look at fished areas, we find that overall there's less parasitism in fished areas, as indicated by the red lines, than there are in unfished areas. So by entering into this food web, we can initiate trophic cascades that have indirect effects on parasites, and those indirect effects are critically determined by the presence of one crab. If we took the crab out of this food web, the effects of fishing would be the opposite on parasitism. Fishing would increase parasitism. I wanted to end with one last example of why that it's more than just theoretically interesting to study predation effects on parasites. And that has to do with this human infectious disease, schistosomiasis. This is a disease that is historically and in some places still quite prevalent in the tropics. It has it's a trematode-based disease, trematode-like a lot of the other trematodes I've been talking about in this talk. The parasites live in the circulatory system, the veins of a human. There are many types of schistosomes that affect other hosts as well, mammals, birds, and reptiles. But I'm going to be talking about human schistosomiasis here, the schistosoma mannsoni being the primary one of interest here. They have a snail intermediate host, and they do not have a second intermediate host. It's a simple one snail, one final host life cycle. The eggs are produced by worms that live in the veins, and when the adults produce these eggs, they're in the circulatory system, but they have to get out of the human through the feces. And another species, Schistosoma hematobium, it escapes through the bladder, and both occur in this particular system. Now you can imagine getting from the circulatory system outside to the outside of a host is very, very difficult. And so these eggs have enzyme secretion and hooks that allow them to pass through the veins into places like the liver or the bladder. And most of those are then attacked by the host immune system. Only a few get out. But these worms are long-lived. They can live for many, many years in a human. They produce eggs every day. And if most of those eggs don't get out and they're attacked by the immune system, eventually they start to fill up the liver, they fill up the bladder. The immune response creates a lot of intense inflammation, and there's calcification that occurs around the eggs, and people, their livers enlarge, their bladders stop working, they die of liver failure. So yes, it's a horrible disease. We began to study this disease in the Senegal River Basin. What was interesting about this particular location is that before the 1980s, the disease was not really a problem at all. Up here in the north, it's very dry up by Mauritania. The disease was known here in Senegal and other places in Central Africa for sure, but it was not very prevalent here up in the Saint Louis area north of Dakar. But you know, lots of people are always trying to invest in Western dollars in Africa. And so there was a project funded by the World Bank. And the idea was to improve the area for farming around the Senegal River. And one of the biggest barriers to farming in this area was the fact that the tide would come in from the Atlantic Ocean and go up hundreds of kilometers And then that you had seawater in here and you couldn't use it for irrigation. So they built a dam to block the tide from going up the river. And they created massive acreages of sugarcane, which made some of the few landowners in the area very wealthy. And it brought in a migration of many people from the southern part of the country, some of which were infected with schistosomiasis. And when they arrived, they came into a habitat that was also really good habitat for the snail hosts that are the hosts for the disease as well. So the life cycle was rapidly initiated, and it became the most intense outbreak of schistosomiasis that we have on record. And it's still happening today. We've been interested in how, we might be able to do various things to improve the situation. And if you're thinking about predators that eat parasites, you remember I talked about fish that eat cercariae. Well, one possible idea might be to ask, well, what fish are in the system that might eat those cercariae so that they don't infect humans? And that is something that I think would be interesting to explore. It's not what we looked at. Instead, we were interested in the role of freshwater prawns. We knew from other places that freshwater prawns love to eat snails. And at least in experiments, we had shown that prawns and crustaceans like this could reduce snail populations. So here's an example from an aquarium where we have the two species of freshwater snail, and the dotted line shows their population dynamics with no prawns in the tank, and then the solid line shows the population dynamics with prawns. And so you can see that the prawns love to eat the snails. Basically, they keep their populations from growing. And also of interest is that the prawns prefer to eat infected snails over uninfected snails. And in the previous talk, there was some discussion about, well, predators may discern whether a host is infected or not and have different feeding preferences. And so the fact that you have prawns that love to eat infected snails, that's potentially an interesting dynamic. And but the question is, why aren't the prawns doing their job? And the reason is the prawns have a life cycle where, as adults, they breed in the estuary, and then the juveniles walk up the river hundreds of kilometers, sorry, and then will live in the river system for years. And so when you put a dam across the river, the prawns cannot complete their life cycle in the system anymore. We would talk to fishermen, and if the fishermen were older than 30 years old, they said, yes, I remember those prawns. And I remember after they built the dam, there'd be thousands of prawns on the other side of the dam trying to go upstream. And then the prawns, just stopped coming because the dam was a barrier to their life cycle. So the idea was, can we, if we were able to reintroduce prawns to areas like this where people are being infected, this is the village access to the river site. They use this to water their animals, to get fresh water for drinking. They do their bathing here. They do their laundry here, et cetera, et cetera. And so we went to some local villages and we talked to the tribal chiefs, and we offered to do a study with them. And the first thing that we did was we went into the schools, and there's two villages here, and we got all the school kids and we had them give us a sample, and we counted that parasite eggs in that sample from them. And we found that one of the schools had a lot of parasitism. The other one had a fair amount, but not as much. And then we gave all the kids a dose of ParAqua-COST, which is a very effective medication for schistosomiasis. And it kills all the adult worms, and the kids are completely cured of parasitism. They're at least cured until the next day when they go back into the water and are re-exposed, because the medication does not offer any immunity to reinfection. So after we treated all the kids, then in one of the villages, the village that had the highest egg count, we offered to bring in prawns. These are prawns from Cameroon that we were able to ship in. And then we built a pen across that river access site to keep the prawns in. And then the kids went back and swam on a hot summer day like kids will do. Now, we were going to keep track of the snails in the pond. And so what we see is that in the rest of the figures, the purple will be the village where we added prawns. In the village where we added prawns, the abundance of snails was much lower than in the site where we did not add prawns, not surprisingly, but similar to the experiments we had in the aquaria. And also similar to the experience in the aquaria, the effect was much greater on the abundance of infected snails. So the prawns seem to be eating more infected snails even than the uninfected snails. But really the most important thing was what happened to the health of the children after putting in the prawns. Now we knew that the kids were all going to be reinfected because this is well known. But the reinfection rates in the two treatments were quite different. The village that had the highest initial infection rates had relatively low reinfection rates compared to the village where we did not add the prawns. And so that's just an indication that understanding a little bit about parasites and food webs and also the effect of predation on either parasites or on the hosts of those parasites. Not only is it of interesting, academic and theoretical ecology, perspectives, but may be able to be used to alleviate some human suffering from parasitic diseases. I want to add, as exciting as this result is, what we found after many years was that it's really difficult to keep prawns healthy at those locations. The water quality is terrible. And people also like to eat the prawns, and so we eventually, unfortunately, were not able to indicate that this would be a long-term, control strategy due to those couple of constraints. But at least from a more from a theoretical perspective, I think it did show the promise for other such interventions. Well, I wanted to leave time for questions, and hopefully I'll be able to hear you. I'll let you know if I can hear you, but I'm going to end the show now.
Thanks, Kevin. And otherwise, can you hear me? Because I'm a little bit in the phone.
I can, yes.
Okay, because otherwise I can also repeat the question if you can't hear it. So
I'll let you know if I can hear it.
All right. Any questions? Maybe can you check online also?
Yeah, can you hear me?
I can. Thank you. Say your name and where you're from so that I at least feel like I'm there.
My name is Antoine. I'm a PhD working with Denver on associates, as most of the people here in this room. And in your last example, you talk about parasites that basically acted as an invasive species because it appeared in a new area. And I wondered if you look at some parasites that also act as invasive species, what are the impact in the new food webs that they invade, basically? Can you also see that?
Yeah, wow, that's a great question. It's one that we've been very interested in for obvious reasons. And so basically, it's a complicated answer, and bear with me as I walk through it. When an invasive species tend to show up with invasive hosts, right? And so that's how you get most of the time when you get an invasive species. Most invasive hosts don't have many parasites, it turns out. So when you look at, if you take a host, any animal species, and you look at the number of parasites that it has where it's native and the number of parasites it has where it's been introduced, Usually, on average, there's about, let's say, 16 parasites where that host is native. It only brings about 3 of those parasites with it to a new location. It also will pick up another 3 or 4 parasites in that new location. And so on average, invasive species have less than half the parasite diversity where they've invaded than where they are native. Okay, so that's the first point. That may be one reason why they do very well. It's the enemy release hypothesis. On the other hand, another way to look at it is that every time you get an invasive species, you don't just get 1 invasive species. You get 5 invasive species. And so the invasive species problem is much, much bigger than we've counted when you throw the parasites in there. And even though most of those parasites probably have no impact on that native system, there are famous examples where they do have an impact. Right now, we've been studying a population of endangered foxes on a small island off just about 50 miles from here. And their population was impacted by a number of things. They spent millions of dollars recovering this fox population, and then a parasite invaded, probably came in with the people who were trying to recover the fox because it uses an insect as an intermediate host. And these foxes, which had been isolated on an island for 10,000 years, were really majorly impacted, and their population was severely crashed, and we're worried about that parasite spreading to other islands. So right now, it's only on one. So there are many examples like this. I would say the main source in, at least in marine systems, is aquaculture, of people bringing in hosts that oftentimes have parasites and then maintaining those cultivated hosts at high densities, which is perfect for parasites, and the parasites can escape out into the natural system. So yeah, great question. I could send you lots of papers about that if you're interested.
Thanks.
Yeah, he's interested because he also, maybe you can tell a little bit about your previous, because you worked at NEOS with.
Yes, it was not totally innocent because I used to work with David Hirsch also at NEOS, and I worked on also invasive parasites in mussels. So
okay.
what really first ones, yeah.
Excellent. So I will ask you, can you add anything to my
Yeah, it's a really interesting system in Wadden Sea because it's so there's a lot of mussel and oyster banks there, and mussels are infected with 2 really similar parasitic copepods, which are morphology really similar, but are actually two different species that are both invading species, but with different invasion histories. So one that invaded in the 40s, 50s, and one that invaded with Japanese oysters. So when they bring Japanese oyster for aquaculture in the 70s, 80s. And so, and one of these species is a specialist. So also come back to specialists and generalists, but one of them is only a specialist of mussels, and one is a generalist of oysters and mussels. So the latest parasite invaded with the oyster and then spilled over in the mussels in the Wadden Sea. So we saw interactions between those 2 parasites in the Wadden Sea. And same, we also saw basically in the first few years when the parasite invaded, we saw a big crash in the mussels aquaculture populations. But it's still not sure if it's because of these parasites, and then it's kind of coevolved together and it becomes less deadly, or if it was just coincidence, and when mussel died, they just saw a big red worm parasite and just assume it was the cause. So yeah, that's a really interesting system, in my opinion.
Yeah, I'll just add this one point, which is that particular dynamic leads in economics to a fascinating phenomenon called a negative externality. So if you are an oyster farmer and you bring in a parasite that escapes out of your oyster farm, and impacts your competition of the wild fishery or the mussel thing, then that's a big benefit to you. And so we actually find, weirdly, it's hard to put regulations on aquaculture with respect to the parasites that they bring in. Anyway, so I study that particular problem with abalone here, and where it's a big problem.
All right. Thanks. Also you, Anton, for sharing that. Yes. Any other questions? Andre?
Hi, Kevin. Thanks for a great lecture. We have talked today mostly about chytrids and parasites of phytoplankton, which basically reinfect the same host species. So, of course, I know your work very well, and there's a lot of parasites that are actually transferred trophically. So, can you give an indication of how common it is? How many hosts does a parasite on average exploit during its lifetime?
Yeah, there's a group, Jeff Parker and Dan Benesh and folks. I've worked with them on this question about the evolution of complex life cycles and how common multiple hosts are in a life cycle, and what are the situations under which you might have 1, 2, 3, even 4 obligatory hosts in your life cycle. And it comes down to, again, there's no way to summarize it all briefly, but it comes down to a few different things. The first is body size. And so as you move up the food chain, obviously your host gets bigger and bigger. And if that's your ultimate goal, like if you really want to be in a big, long-lived host, which is a great, like the parasite that I showed at the beginning, that big trematode, it's in a big host that lives for many years. That's the perfect place. But to get there, the abundance of those fish, as somebody who's tried to catch them many times, there's one fish every square kilometer, right? You're not going to find that as a little tiny parasite larvae. But if you use the food web, that's a very efficient way to get there, because you can start moving up and use that food web basically as a highway system to get to that ultimate goal. So what that suggests is that parasites that use top level hosts, high trophic level hosts, they get there by adding prey hosts to their life cycle. We normally think about the opposite, that parasites might add predator hosts to their life cycle because they're making the best out of a bad situation, right? If their prey host is eaten by a predator, they might as well try to infect them, because otherwise they're going to die. So those are the two routes to add trophic transmission, which is just one of many types of life cycle complexity. And so, that's kind of the main thing, is that it has to do with a route to get to the host that you really want, which is a big, long-lived host, and allows you to become bigger and longer-lived yourself. That's one of the current theories out there.
So do you think that the multi-host life cycles are more common than single-host life cycles?
Well, yeah, that's a good question. You'd have to do it taxon by taxon. So in the helminths, if we were to look at nematodes, there are many single life cycle parasites and many multi-host life cycle parasites. I would say, I'm just going to make a completely uneducated guess, it's probably like 50-50 as far as parasitic species go. If we go to the cestodes, it's like 98% multi-host life cycle, with the majority of those being 2 or 3 host life cycles. If we were to go to acanthocephalans, it's 100% complex life cycles. When we talk about protozoans, it's probably more like 15% complex host life cycles. I'm just making wild guesses because you're forcing me to, but I think it would be a really interesting study to make those calculations if it hasn't already been done.
So maybe as a follow-up question to that, I was wondering, So with this increasing complexity, so to say, with increasing number of hosts, so to say, the parasites will rely more on longer term survival, right? So they need to survive in different hosts. So do you see in these parasites also more like dormant stages or protection, or like any ways that they can survive the time to really reach their, well, the highest profit level they can get to?
Yeah, so there's, well, first let me point out something that you were alluding to, which is that when we do our food web analyses and we ask which species are the most sensitive to a food web becoming simplified, what species are going to be most likely to go extinct? It's the parasites with complex life cycles that go extinct first. Because, you know, it's bad enough to be completely reliant on one host species, but if you rely on 3 or 4 specific host species to be present to complete your life cycle, you know, that's a lot of things that have to go right for you to be able to persist in the system. So complex life cycle parasites, in some ways, it's hard to imagine why they should evolve, because they're so sensitive and they require complex functioning food webs. Indeed, we find that areas with complex functioning food webs have a higher diversity of parasites with complex life cycles in them. So that's just one point that to make. So you might ask, why evolve complex life cycles in the first place? That's another question, what we just talked about previously. But then the question about, do parasites have other types of stages, or, you know, let's say insurance, so to speak. And some do, but not as much as you would expect. Like not the sort of stages that you see, for instance, zooplankton, like with Daphnia, where you have this resting stage that can be in the sediment over the entire winter. I mean, the parasites do have egg stages that are not metabolizing much, and they can stick around for a while. But we don't see those, I would say, really impressive adaptations to persisting outside of a host for a long period of time. Their time is limited. The clock is ticking for them to get into a host. Once they get into a host, they can live for a long time. Surprisingly, those schistosome worms that I showed at the very end, they may live 25 years in a human. So once you get there, it's great, but your odds of getting there are low.
All right, thanks. So maybe again, if there's no hands raised, and I just take this opportunity. Because the other thing, you showed quite a few things, right? Like making these estimates of contributions of the parasites to the system. And now we just talked shortly about this, like the time scale for the whole infection process across these different trophic levels. And we talk here also about scaling, right? So it's one of the things we also want to touch upon a bit more tomorrow, and the different talks touch upon that. So how do you deal with this time scale aspect in terms of when you go in the field, when you do your assessment, and how may these estimates in terms of the impact of parasites depend on when you are in the field? Especially in well, maybe not in the tropics, maybe less than in temperate regions where you have these strong seasonal dynamics, etc.
Sure. Yeah, I'll give you 3 examples from 3 systems that I talked about. So in Palmyra, in that coral atoll, it's all the same anytime you go. It's just always wonderful.
Yeah.
And so it doesn't matter. In California, if you go in the middle of the summertime and you do a zooplankton tow, you'll find a lot of trematode cercariae in that tow. If you go in January, you won't find any. So there's a lot of seasonality to the production of those infectious stages that's limited to warmer periods, primarily because in that system, the production of infectious stages is tied to the snail food dynamics, the food budget of the snail. And in the winter months, there's just not enough light to support the periphyton that the snails feed on. So that's a seasonal dynamic. If we go up to Takvatn in northern Norway, you know, there's ice covering that damn lake for 9 months of the year. So yeah, you're pretty limited to when you're going to even be able to do work. And things go quiet for most of the time. And it's a very short period where, you know, all those animals have to do their thing in basically 4 months, I think. So now how that affects host parasite dynamics, I think it's really an open question, a fascinating one. And I know that in the chytrid system, most of the work has been done in these incredibly seasonally dynamic lake systems. And so much of what the papers that I've read and the way that I've thought about the system is very much exactly like you described. It's a seasonally pulsed dynamic that follows some patterns. If you were to go to the Amazon, where of course we have chytrids, or other places, you know, would the system be different? And I suspect that's not be a great question for modeling, really, to start with, and maybe some empirical work. So the answer is I don't know, but it wouldn't surprise me that we have strong seasonality in any sort of food web dynamics, including chytrids or these other things that I've talked about.
All right, thanks so much. So, yeah, Serena.
Yeah, I just have a short question, Kevin. Thanks a lot. I was wondering about dormant stages. I mean, is there a way they are taking into account in food webs, or just something that it stays on the side for a while and then just come back?
Yeah, I think that particularly if you build a simple food web model and you allow a stage to be dormant, it must change the dynamics radically. And you know, you could ask that guy next to you, Andre, what his thoughts would be. Right? I mean, it's just you know, that sort of delayed time and the ability to sort of persist doing nothing changes the dynamics a lot. And in some ways, you would expect it would be stabilizing, but in other ways, you add a time lag to things, and it can become, you know, add to a lot of odd feedback. So I don't know if I answered your question, Serena, but I think it's an interesting topic that is quite relevant to these phytoplankton, zooplankton systems that you guys are thinking about.
Thank you.
Any more questions? I mean, we take a bit more time because we have time in the schedule, and I thought I mean, maybe, I don't know if you want to go back to sleep or not, but we just take the opportunity now just while you're here. So is there any last questions maybe?
Yeah. Hi, Kevin. It's
Hi.
Joe here. I really like the idea of you finding those limited diversity landscapes to sort of model food webs on. I thought that was very interesting, and sort of turning a negative into a positive where you haven't got that many species. So let's see if we can do some sort of mathematical mapping. Are there any other sort of environments that you've got your eye on that could be good examples of sort of very basic food webs? Like I was thinking about at high altitudes, you know, low species diversity, and I saw it also that you had that atoll. That was another very interesting idea.
Yeah. So, I mean, firstly, to be honest, I think the most exciting one is the one you guys are talking about. You know, high altitude lake systems or you know, latitude lake systems. Even though they are complicated in their own way, they're tractable, I think, in many ways. I like to work on islands because they're constrained. You know when you're in and out of the system, and you can do sort of a full accounting of the species that are there. That's useful. And other types of things that are like islands. So an estuary is kind of like an island, right? You know when you're in an estuary and when you're not in an estuary because your boots are muddy or they're not. But you know, we've thought about, well, what about places like the deep sea or you know, other things, and for us, to be honest, like if it's an interesting place to go, if we could have an adventure there, we'll go. Like that's the most important thing, more than the tractability of the system for doing science.
And it's nice and close to your university, as you said.
I'm sorry, say it again.
And it's nice and close to your university, the estuary.
Yeah, that's
so
right.
That's helpful as well.
Yeah, I will say, and this won't surprise any of you, but almost everything I talked about was never funded by a grant agency, right? I mean, you know, this is not we're not out there curing cancer or anything like that. But so yeah, we do try to do things that we can do inexpensively. I wanted to say to you guys, I really wish I was there. I love this system. It's fascinating. If there are ways that I can be involved, don't hesitate to reach out to me. And really, again, I am so sorry that I can't be there. And hopefully, there'll be a chance in the future to make up that plane ticket.
Yeah, because we still have that, and yeah, we're also sorry you, yeah, especially for you not being able to travel here, and so we still have the ticket for a year, so we keep that, and we'll see how things go. So thanks again, Kevin, for joining.